Assalamualaikum and very good morning. Today we will go through to the chapter 8, Recombinant DNA Technology. For learning outcome, there are two learning outcomes. First, I will define recombinant DNA technology. Second, I will define and explain the tools used in recombinant DNA technology that consists of target DNA, restriction enzyme, DNA cloning vector, host cells, and modifying enzyme. So for introduction, what is meant by DNA technology? DNA technology refers to the main techniques for sequencing and manipulating DNA and for using these DNA tools to analyze gene expression. What is genetic engineering? Genetic engineering refers to the modification of the DNA of organism and to produce new genes with new characteristics. Refer to this diagram, we can see the genetic engineering applied in animals such as sheep and pig. And from genetic engineering, the term for gene cloning exists. Gene cloning refers to the production of multiple copies of single gene, and gene cloning have two basic purposes. First, to make many copies of a particular gene. Second, to produce a protein product. Next, I will explain about the tools used in recombinant DNA technology that consists of target DNA or gene of interest, restriction enzyme, DNA cloning vector, host cell, and modifying enzyme. First is target DNA. Target DNA refers to the sequence of DNA containing gene of interest, or cDNA. For example, cDNA for insulin production. And the sources for target DNA, example in human, animal, and plant. Second tool is restriction enzyme or restriction endonuclease. Restriction enzyme refer to the class of enzyme that recognize and cleave or cut DNA at specific base sequence. Each restriction enzyme is very specific. The function of restriction enzyme are recognizing a particular short DNA sequence or restriction site and cut both DNA strands at precise point within restriction site. There are three examples of restriction enzyme. First, EcoR1 that recognize the GAA-TTC for its restriction site. So this diagram shows how the restriction enzyme cut the restriction site. For EcoR1, it will cut between G and A at 5 to 3 prime, and the opposite strand, it will cut between A and G at 3 and 5 prime. Second example for restriction enzyme is BAM-H1 that recognize 5 prime GGA TCC 3 prime. Two main types of restriction enzyme. First, cut DNA fragment forming blunt end. Second, cut DNA fragment forming sticky end. In this diagram, we can see the straight cutting that form blunt end. And in this diagram, we can see the staggered cutting. This slide shows 4 examples of restriction enzyme that forming blunt end. First, SMAR 1 derived from Serrana marcescens. That recognition site is 5' CCC GGG 3', and the opposite strand 5' CCC GGG 3'. Second example, POV2 derived from Proteus vulgaris, and the recognition site is 5' CAG CTG 3', opposite strand 5' CAG CTG 3'. Third example is HE3 that come from Haemophilus aegyptius. The recognition site is 5' GG CC 3', opposite strand 5' GG CC 3'. And the fourth example is HPA1 that come from Haemophilus parainfluenzae, and the recognition site is 5' GTT AAC 3', and the opposite strand 5' GTT AAC 3'. The third tools that we use in recombinant DNA technology is DNA cloning vector. DNA cloning vector refer to a vehicle used in gene cloning to insert a foreign DNA fragment into the genome of a host cell. It can be cut with same restriction enzyme as the foreign DNA fragment. There are 4 types of DNA cloning vector. First, Plasmid. Second, Bacteriophage. 3, Cosmid. And 4, LECT, yeast artificial chromosome. So this slide shows the type of DNA cloning vector. So this is Plasmid. This is Bacteriophage. This is Cosmid, combination Plasmid with Bacteriophage, and this is yeast artificial chromosome. Next, what is Plasmid? Plasmid is small ring or sequela of double-stranded DNA molecule that carry genes separate from the main bacterial chromosome, and they replicate independent from the chromosome that carry foreign DNA into a host cell and replicate there. First type of DNA cloning vector is Plasmid. Example, PUC18. So Plasmid are small, usually carry only one of a few genes and have a single origin of replication. It's easily isolated from a bacterium, and they can be reintroduced to another bacterial cell by transformation process. Its capacity to carry insertion size 10 kilobases. Second, bacteriophage, example lambda 2001. As an alternative technique, bacteriophage can also be used to introduce recombinant DNA into cell. These viruses have been disabled so that they do not cause disease in the cell to which they are introduced. Instead, their DNA and the foreign DNA that they are carrying are incorporated in a chromosome of the host cell. This mechanism is most useful in attempts to clone larger genes. The capacity to carry insertion size 15 kilobases. For Cosmid, example SCOS1, Cosmid refer a hybrid vector that used for gene cloning that includes the source gene, source site that from the lambda bacteriophage. Hybrid vectors between plasmid and lambda bacteriophage chromosome. It also contains drug-resistant marker genes and other plasmid genes. Cosmid can incorporate larger DNA fragment than either phage or plasmid vector alone, suitable for cloning large mammalian genes or multi-gene fragments. Capacity for Cosmid is 44 kilobases. Next, yeast artificial chromosome, example PYAC, that refer to the vector that is modified plasmid used for cloning large segment of DNA in yeast cell. This chromosome, like vectors normally in mitosis, cloning the foreign DNA as the yeast cell divide. YAC or YAC can carry a much longer DNA segment than can a plasmid vector. A clone fragment is more likely to contain an entire gene rather than just a portion of it. The capacity to carry insertion size is 500 kilobases. The fourth tools that we use in recombinant DNA technology is host cell. Host cell refer to the organism that has been used to receive a recombinant DNA for cloning purpose. The organism that can act for this function is bacteria, example E. coli and yeast. The last tool that we use in recombinant DNA technology is modifying enzyme. Example, DNA ligase. That the function of ligase is rejoin the cut or broken DNA strand. We use in DNA repair and recombinant DNA. Function of modifying enzyme. DNA ligase is repair enzyme that catalyze the joining of broken parts of DNA. Once two molecules have been joined together in this way, they can be treated with DNA ligase. DNA ligase used to covalently bond the pair ends of the plasmid and human DNA, forming recombinant DNA. Next, tag polymerase. A special heat-resistant DNA polymerase uses nucleotides and primers to replicate a DNA sequence in vitro, thereby producing two DNA molecules that use in PCR or polymerase chain reaction.